Thank you everyone who's coming in so far for joining. So welcome to our webinar, Cracking the Code, Genomic Research on Nephrotic Syndrome. My name is Destiny. I'm NephCure's Content Marketing Manager, and I'm joined by the wonderful Dr. Matt Sampson, one of NephCure specialists and pediatric nephrologist, who I'll actually let introduce himself, his occupations and work, and I'll let him kickstart our webinar. But if you guys have any questions during the webinar, please feel free to drop them in the chat and we'll answer them during the last few minutes of the webinar while we have our little open Q&A towards the end. So Dr. Matt Sampson, I will let you kick us off.
Great. Well, thank you so much for the introduction and for the invitation. And it's always a great opportunity to share my some of my thoughts on one of the passions that I have in my life, which is we're working on cracking the code and using genetics to help discover causes of nephrotic syndrome towards treatments and cures. I'm a pediatric nephrologist at Boston Children's Hospital and affiliated with Harvard Medical School and the Broad Institute and the Brigham and Women's Hospital. And I always knew I wanted to be a pediatrician, but thought I was going to be doing well child checks and sore throats. But I became really intensely interested with my head and my heart about nephrotic syndrome. And as the years have gone by, in my practice, in my research career, I've shifted to almost exclusively research. So having the opportunity to talk and share our work, especially with patients and families in venues like this and at the Patient Summit in a couple of weeks, is really special to me. So thank you so much for the invitation. Like Des said, feel free throughout the talk, if you have any questions, to put them in the chat. Or if you have the ability to raise your hand or just speak up on this, I'm not sure what the restrictions are, you can feel free to do that. I'm going to take the next 20 or 30 minutes to describe a little bit of what we're doing in the Sampson Lab for Kidney Genomics. And I just want to introduce Waverly Alcure, who's our social media coordinator, community outreach coordinator in the lab. And I thank her very much for putting some of these slides together and being my partner in crime on this. Okay, so what are we talking about with nephrotic syndrome? So nephrotic syndrome is a disease of the kidneys, and here's a cartoon of the kidney in the upper left-hand corner. And most of us have two kidneys, and each of our kidneys has about 1 million filtering units in each of them, and these are called glomeruli. And this is the big cartoon of a glomerulus. And what the glomerulus does is, the way I explain it to families is it acts like a spaghetti strainer or a coffee filter. What you have here is you have blood coming through the you have blood coming from the body and bringing in red blood cells and proteins and waste and fluid, and the blood flows through these capillary loops, and then the blood returns to the rest of the body, so it leaves the glomerulus. And in the meantime, waste products cross these capillary loops and go into this space called the urinary space or Bowman's capsule. So essentially what I say is that the glomerulus' job is to keep the good stuff in and let the bad stuff out. And nephrotic syndrome, or any proteinuric kidney disease or protein spilling diseases, is a disease of the glomerulus when these filter units don't work as well. And what happens is these structures become a little broken. So I want to show you a real life picture. This is an electron microscopy. And now if you can imagine, we have one of these red capillary loops here. Now pretend we're floating in a little spaceship around the outside in this little space, and we're looking down at the capillary loop. And this capillary loop is being covered by the cells that look like little octopus arms. And this is the cell body. And these are the primary processes, and these are the foot processes. And these are cells that hug the cap, and they form a barrier by their size and their charge to only let bad stuff out and keep the good stuff in. Well, in nephrotic syndrome, this beautiful architecture goes away, and you lose the architecture, and you can see all the foot processes flatten out, and it's called podocyte effacement. And when this happens, all of a sudden your glomeruli lose tons of protein, which include clotting factors, immunoglobulins, etc. And the blood that goes back into the body no longer has protein in it, so there's swelling. And this is on the microscopic level, is what underlies nephrotic syndrome, FSGS, membranous nephropathy, and all these other primary proteinuric kidney diseases. So the problem with nephrotic syndrome as a clinician taking care of patients is it's very easy for me to say, oh, your child has nephrotic syndrome, or you have nephrotic syndrome. But the way we describe it currently is very descriptive and it's imprecise. We can characterize someone as being if we do a biopsy, and this is a picture of a biopsy, and if there are minimal changes on the biopsy, this is a glomerulus, we say you have minimal change disease. Or if there are focal areas of segmental sclerosis, we say you have FSGS. At the same time, for kids especially, we give them steroids or other immunosuppressants, and if they respond to steroids, they have steroid-sensitive nephrotic syndrome. And if they're resistant, we give them the classification steroid-resistant nephrotic syndrome. And I think you all can appreciate that SSNS, SRNS, steroid-sensitive, steroid-resistant nephrotic syndrome, minimal change, FSGS, these are all descriptive. They allow us to give a patient a diagnosis, but it gives us no understanding of the molecular anatomy or the biological changes that are causing this disease. And in order to make medicines and work towards cures, we really need to understand mechanisms. And furthermore, if we know that there are many different molecular mechanisms or biologic factors underlying MCD, it's really important for us to be able to tell this to our patients. Because if I have in the course of a year, if I take care of 40 patients with minimal change disease, there may be 10 or 20 different biologic mechanisms that cause minimal change disease. And those mechanisms may have totally different outcomes. And this is the concept of precision medicine, which is getting at the details, the molecular causes and contributors to these conditions. And we know that we're doing okay, and I think decades ago, before antibiotics and other therapies, a lot of people died of nephrotic syndrome, and thankfully that doesn't happen anymore. But we can certainly do better, because right now many patients with nephrotic syndrome suffer from chronic kidney disease, or they go to end-stage kidney disease, ESRD. And some patients even have a recurrence of their disease after they get transplanted. And some of the medicines that help people get into remission at the same time cause people to feel very sick. So there are lots of side effects. So we need to do a better job to reduce or even prevent patients with nephrotic syndrome from having CKD or ESRD, and to design medicines that work really well to induce remission and don't cause all the side effects that those of us who know patients or who themselves have been on steroids or Cytoxan or Stelcept suffer from themselves. So what are we doing in the Sampson lab for kidney genomics at Boston Children's Hospital? So what our lab is doing is we're working towards genetically informed treatments and cures for nephrotic syndrome. Let me tell you a little bit what that means. Essentially, what it means is, we want to get here, and I have just a big X because this is just a non-specific cartoon. We want to get here, and by here it means, knowing the genetic causes and knowing the molecular mechanisms and designing the medicines. And where we are today is here. And if I made this table, this slide bigger and extended it to the left, we've made a lot of progress. I came into this field at about 2010 as a researcher, and for 20 or 30 years, people have been working on it. But I came in this situation, or 10 years ago. Now here in 2023, we have a lot of activities to work on here, and I've put them in green lines and red lines and yellow lines, and they're different lengths. And what that means is that, if our goal is to get here, there are some projects that are denoting green that are more easily able to get to, and the shorter the line means that we're getting even closer to our goal. In yellow are the intermediate projects, and then in red are those that are harder, that require a lot more effort and money and time. And some of those we've been working on for a while, and we're almost there. And others, we're just at the beginning. So an exciting part of the research that our lab is doing in collaboration with others in the United States and around the world is working on projects that are, we can see the end on the horizon, others that we realize are now important and we need to get to work on. So let me talk about genetics for a second, okay? So for those of y'all who don't think about genetics as much as me, which I would guess is almost all of you, because I think about genetics like 60 hours a week, the genome is the scientific term for all of your DNA. So let's say this is a person with nephrotic syndrome. What they have is they have half of their DNA from their father and half of the DNA from their mother. And combined, these DNA sequences encode your genome, which encodes mRNA, and then that makes proteins. And proteins are what is the major contribution of your cells. And your cells come together form tissues and organs. And what your genome does is some things like that's much more obvious for folks is, your genome can influence your height or your eye color. So there's genetics that if you have blue eyes, family members are more likely to have blue eyes. But genetics, especially when genetics are abnormal, they can also lead to disease. So patients who have nephrotic syndrome, I would say that 100% of them have a genetic contribution to their condition. Now, that's a statement, and if I just left it there, it would lead to a lot of confusion. But what we know is your genetic code, you have about 3 billion letters in your genetic code. And for I would say about 5% of people in the United States with nephrotic syndrome, one letter change in your genetic code is sufficient to cause the disease. And that's called a Mendelian form of disease. For the other 95% of people with nephrotic syndrome, so the vast majority of people with nephrotic syndrome in the United States, including essentially every patient who responds to immunosuppression, those people, they have changes in their genome that increase the risk of getting the disease, but are insufficient alone to cause it. So for people like that, they have a genetic change, but then they need to have some sort of exposure to a virus or an infection or a collection of different genetic changes to cause the disease. And what we spend all of our time doing in our lab is trying to map these genetic changes and try to figure out the genetic changes that contribute to nephrotic syndrome. So in a little bit of a cartoon, the model that we use in our lab that I share a lot, if you've heard me speak before, you've probably seen our gumball machine, is that in my mind, I said there are 3 billion base pairs in the genome or letters of the genome. And here, these gumballs, each of them represents one letter that when altered contributes to the nephrotic syndrome. And our job in step one is to use computers, statistics, patient blood samples, saliva samples, DNA samples to expose one of these genetic changes as a cause or contributor to disease. And I have this represented as a purple X. And that takes a lot of work. It's a lot of math, a lot of money. It's a lot of involvement from patients in research. But if we can find this purple genetic change, we could do two things. One is we could do clinical association studies, which is called epidemiology. So we could say, oh, everyone with nephrotic syndrome who has the X change, they're younger when they get it, or they have a lower kidney function when they get it, but their outcomes are better. Or if they have a different letter, then they'll have different clinical associations. So this could be really important because you can imagine if you went into clinic and you or your child, we diagnosed your child with nephrotic syndrome and we said it's due to this purple X change, we could say we've done a lot of research and we know patients with nephrotic syndrome with that purple X nephrotic syndrome, they have this kind of baseline characteristics, this kind of response to therapy. So that's using a genetics as a biomarker. But I'm greedy, and most of my friends and colleagues are greedy. We don't just want to have biomarkers. We want to make drugs that cure the disease. So we could also take this purple X, and we could define the biology and the mechanisms that this genetic change causes. And if we could identify the mechanisms, then we could make a drug. For instance, one of the most common things that will happen with a genetic change is it turns a gene off. So all of a sudden, people have a deficit of a certain gene. So if you guys were all unmuted and you were my medical students, I would say to you, here's a quiz. If you suffer from a deficit of a gene, a protein because of this, what's the drug? And all of you would ultimately answer, well, we'd give a drug that mimics the protein that's lost. At the same time, if this genetic change causes overexpression of a protein, then we give a protein blocker. And that's essentially the foundation of what we do in the Sampson lab. So we use existing technologies and methods when appropriate, because I'm not looking to develop new technologies or develop a new method if one that is already there is good enough, because I want to focus on finding answers. I don't need to worry about making tools. We take advantage of emerging tools when they're available, but we also have the ability and expertise to develop them when we need to. And finally, our goal is to share our data as openly, as usefully as possible. Most of our data comes from patients and families who volunteered their samples. and volunteered their time to be part of research. And I am very lucky to have been trained by great mentors. I've worked in amazing institutions at University of Pennsylvania, Children's Hospital of Philadelphia, University of Michigan, now at Boston Children's and Harvard Medical School, where I've had the opportunity to learn and now execute the building of lots of data from patients with nephrotic syndrome. And my philosophy is not to keep that data locked away in my drawer for only my benefit. It's to take advantage of our ability to share and the consent from patients to let people from all over the world, not just America, who have the expertise, but maybe not the opportunities that I've had, to use the data that we've generated for the benefit of all patients. That's been really, really meaningful for us. So where do we want to get ultimately? And I've alluded to this. We want to discover the genetic causes of nephrotic syndrome, causes or contributors, the molecular mechanisms, and ultimately design a treatment and a cure. And there's actually a step farther than a treatment and a cure. And again, if you were on my team, I'd call on you and say, what's even better than a treatment or a cure? And someone would say prevention. I think that would be really, really cool and excellent. And I do think that within my lifetime, um, which hopefully will be another 50 years, as long as I get back to running and eating better, I do think that we're going to be able to figure out in some proportion of people to prevent them from getting a disease, because we will know their genetic signatures and we will know what to do to prevent. And we will certainly end up having cures for many of these forms of nephrotic syndrome, as well as treatments. In addition to this, I always say that if I'm the guy giving these talks in 10 years from now or 5 years from now, if I'm the only person talking about this, then I've failed, because I need to, and we as a community, people of my vintage, we need to train the next generation of kidney genomics or kidney omics investigators. And we also need to move away from just having, cis white males being dominating biomedical research for nephrotic syndrome. There are plenty of people who have not had opportunities before, who've been historically underrepresented in medicine. And I think we've been doing really well using strategies to get folks from these underrepresented groups to become the next generation of physicians and scientists, investigators to contribute. So this is a little bit of at our lab. We have a very large and diverse group in terms of expertise and demographics, led by myself and my co-lead investigator, Dong-Won Lee, who is originally from Korea, and he is a computational genetics guy. So me as the physician scientist and Dong-Won is the computational guy and statistician. We are able to bring together a group that includes folks like Michelle, who's a statistical geneticist, and Anna, who is a adult nephrologist, the Brigham, and a genetics expert. Kathy, our clinical research coordinator, and Anya is our computational biologist. AC and Jane Witt are also both clinicians as well as scientists. Waverly is our community outreach and social media coordinator. Iman is one of our research interns. She's a Harvard undergraduate. She's working on projects. And Somya and Junmo have recently joined Anna's group, and they are computational folks. Jirthi and Eric are both clinician scientists. Jirthi has a master's degree in bioinformatics, and Eric is an undergrad who's going to be going and getting his MD PhD. And then Adrian is a pediatric nephrologist, and Dave Vanderwall is a Harvard medical student who's volunteering his time. So you can see we've a diversity of expertise and a diversity of backgrounds. So we're all bringing to the table both shared expertise, but a lot of unique expertise, united by this common goal to map the genetic basis of nephrotic syndrome towards treatments and cures. And it's a fun atmosphere. It's a really fun group, and we take our work very seriously, but we don't take ourselves too seriously. So our mission is really fourfold, and it's to discover I've talked a lot about this already. We want to contribute to the development of genomically inspired treatments and cures. We want to educate. So, genetics is relatively new and it's not covered a lot in medical school, and a lot of folks don't know about it, both doctors and families. So we want to increase the genomic literacy of adult and pediatric nephrologists to help them use genetics, genetic medicine, which is not only just medicine, but also genetic care of patients. We want to connect families and children affected with nephrotic syndrome with resources and support and the research that we're doing that could help contribute to their improving their health. I know we really started this outreach about a year or two ago with Waverly, and I had a belief that there are people out there um, who would really be interested in what we're doing scientifically, but also just emotionally engaged in our work, knowing that there are people out there fighting for what can often feel like a disease that no one else in the world knows about or has. So I just wanted to share two quick stories about some of the work that we've been up to. So one study we've recently been doing is mapping the genetic basis of children with steroid-sensitive nephrotic syndrome. So that represents about 80% of children in the world respond to either steroids or tacrolimus or calcineurin inhibitors or CellCept. So what we hypothesize is that there's a genetic change that is in these children that is not present in healthy children. And we want to find that. And what we did is we did a worldwide study with patients from Europe, like France and Italy and Poland, from South Korea and Japan, from India, from Nigeria, from North Africa, and Hispanic individuals. And we did a genetics study. And this is a map of our genetic results. And every time you see a little tower here, we call it a Manhattan plot, like the skyline of Manhattan. Every time you see a little tower, that means that's a part of the genome. And on the x-axis here are those 3 billion changes. And every time you see something above this red line, that means we found something. And once we found these, they're called loci. We then integrated our genetic information with cellular information and immune cell information. We did a lot more work to try to go from genetic variant to function. And what we found thus far is 12 risk regions of the genome. 8 of them are new. And we are starting to implicate specific genes. And we're starting to map those genes to specific cell types. And as I said earlier, patients with immunosuppressant sensitive nephrotic syndrome often have a problem of the kidneys, but often it's their immune cells that are messed up too. So this is getting at genetics to molecular mechanisms. And if we could figure out like a gene that we've been studying, like CalHM6 seems to be overexpressed T cells, then we can work with our colleagues to study T cells and CalHM6 a little bit more. So I'd be happy to answer more questions about that, but we're starting to make progress on finding genetics of this condition that we had never found before. Some other work we're doing is on APOL1 mediated nephrotic syndrome. Mostly this is FSGS. And what we've known for a long time, we've known for a long time that Black individuals have a higher incidence of kidney disease. And we have about 10 or 11 years ago, we identified, not me, but our colleagues identified this APOL1 gene that has a couple of abnormal genetic changes that greatly increase the risk of nephrotic syndrome in these individuals. And while in the US population, people who self-report themselves as Black, 13% of them have this risk factor. Those who have FSGS, 70% of them have this risk factor. So lots of folks have been doing work to figure out, why does APOL1 cause kidney disease? And that matters a lot, because if you think about it, if we could figure out the molecular mechanism of APOL1 kidney disease, then we can make a medicine and perhaps cure or prevent the disease percent of Black people who have this condition. So we've done studies, and we've looked at we've compared Black patients, self-reported Black patients with kidney disease who had or lacked the APOL1 genetic change. And we found that if you have the APOL1 form of nephrotic syndrome, you have a more severe condition. Your kidney function is lower at baseline. You have more scarring on your biopsies. There's less of a chance of achieving complete remission and a faster decline of kidney function. And furthermore, do I have it here? No. So in addition to knowing that APOL1 kidney disease is more severe, other folks have gone after the mechanisms, and there's now drug trials that have been going on, including one that was recently published in the New England Journal of Medicine that has an APOL1 specific therapy. And this is one of the first gene-specific therapies for nephrotic syndrome, and our hope as a community is that this is the first of many that allow us to be more specific and more precise in caring for patients based on the genetic knowledge of the condition. So moving forward, our kidney omics approach for nephrotic syndrome in the Sampson lab, we want to recruit more patients and controls. We want to generate more data sets from patients' DNA as well as their profiles of their kidney cells. We want to do some more screening using experimental work, and that will help us accelerate and characterize the function of molecules. And we want to take advantage of lots of technologies that exist now, including single cell biology using stem cells and organoids. And finally, we want to contribute to launching a therapeutic development program. I don't think at this point my lab is going to develop a drug, but what happens is labs like mine and our colleagues, we work with biotechs and companies that partner to help create drugs of interest. And when their opportunity arises, we want to have enough money to invest in opportunities as they come up. So if there's a new technology or a new data set, I want to be able to say, hey, I think this is going to really help us with Genomics research, and we want to go after it. So Waverly's really led our work on connecting with the nephrotic syndrome community. We launched our social media in October of 2022, and we have a Facebook and an Instagram and a Twitter and TikTok, and we have 900 total followers. We've reached 62,000 users via Facebook and Instagram, and we've had 143 impressions on Twitter. And we're really hoping to raise awareness about nephrotic syndrome in our research to discover, the drivers and potential treatments of the disease. And it's been really cool. Waverly and myself, we've interacted with folks in the US and around the world on multiple continents who've reached out to us and say, thank you for sharing your work. It's really interesting. Or how can I be involved? Or can you send me more information? And we've been able to, provide more information, maybe a little bit more hope and a little bit more engagement for folks who are really thirsty for this kind of stuff. And finally, I always bring up, for folks who want to have the opportunity to participate in kidney genetics research for proteinuria kidney disease, we do have our Big Kids study. And this is a study where we analyze the genes and genetic variants and how they contribute to nephrotic syndrome to contribute to personalized medicine and identify the genetic causes of the disease. So we've made our Big Kids biobank that holds health information and biosamples from patients both at Boston Children's and in the US and abroad. And if you're interested, here's a QR code. You could also email this address. Right now we just enrolled our 210th patient, and we are looking forward to digging in and hopefully finding more causes towards treatment and cures. So with that, I'd just like to thank everyone, and happy to spend some time answering questions and talking about how we can accelerate genetic discovery together. So I'll stop sharing and happy to take any questions.